In the last two videos, we saw the cell cycle controls, and DNA replication. So once the cell reaches a point in its cell cycle where it's ready for cell division, and it has successfully replicated its DNA in the S phase, and it has successfully met all of the checkpoints, and the G2 checkpoint tells the cell that it's all ready to go, the cell is going to enter a process of cell division. Now, there are two types of cell division. The first is mitosis, and we are going to talk about what occurs in mitosis and why mitosis occurs. In the next video, we will talk about meiosis, which is a specialized type of mitosis that is only for the production of sperm and eggs, so our reproductive cells. So mitosis basically has one job. This is to generate new cells that are genetically identical to the parent cell. So this happens for two reasons. One is growth and development, the other is replacement. So when I was talking about DNA replication, I stressed the requirement for cells to pass on the correct genetic information. Now, if you're a skin cell, you want to make a skin cell, right? You want to pass on the information so that that cell can function as a skin cell. And that's one of the jobs of mitosis, is to replace existing cells. So as an organism grows and develops, it gets bigger, and that growth and development happens because new cells are produced. So a plant root, for example, grows about an inch a day, or I'm sorry, half an inch a day, sometimes faster, depending on the plant, and it is one of the fastest growing parts of the cell. And the reason is there's a particularly rapid bit of cell division going on at that root tip. All of these cells are dividing, causing the root to grow longer and longer and longer. The other job of mitosis is replacement. So skin cells have to be replaced, or I'm sorry, cells in general have to be replaced when they die. Basically, wear and tear from living causes damage to our cells. So for the example here is shaving. Every time you shave, it damages some of the skin cells. Those skin cells have to be replaced by the ones that are still there. Most of those are dead anyway, that's why they're on the outer layer, and so shaving basically removes those cells and provides a new place for the cells that are still existing to divide. So there are other reasons that cells might need replacement. If you get a cut, you have to repair the skin damage. Other sorts of damage, say you have you get a scratch on your eye, or an ulcer or something inside your stomach. All of those are reasons for replacement. So just as a recap, the job or the purpose of mitosis is to produce new genetically identical cells. So mitosis occurs in a type of cell called somatic cells. These are basically all the cells in our body except for our gametes, which are sperm and eggs. So once we are done growing, mitosis basically functions to replace worn out cells. Now, the rate at which this occurs depends on the organism, it depends on the organ, the location. As we said in cell cycle, or as I said in cell cycle, some cells never reproduce. Brain cells don't reproduce, heart muscle tissue doesn't reproduce, liver cells reproduce very infrequently. Others reproduce much more rapidly. Red blood cells, for example, are replaced every 2 to 4 months when they wear out. Skin cells are replaced every few days. The lining of our intestines are replaced every couple of weeks. I think I said 1 week in an early video, so go with 3 weeks. And when they are too worn out and maybe their telomeres are all used up, or just generally can't function anymore, the cell will undergo something called apoptosis, which is sometimes called cellular suicide or programmed suicide, programmed cell death. It's basically controlled cell death when the cell is no longer capable of dividing and reproducing. It'll essentially stop. But remember, this is important because after too many rounds of cell division, the telomeres are degenerated, and the cell is no longer able to pass on the proper genetic information. So let's talk about the process of mitosis. So mitosis basically produces identical daughter cells. So we're going to start out with a parent cell. We're going to go through interphase, meet all of our checkpoints, and then the process of mitosis. If I say meiosis in this video, I mean mitosis. I'm going to try not to, but just in case, we're talking about mitosis right now. In the process of mitosis, the chromosomes pack nice and tightly, they line up neatly, they are separated, pulled apart, and partitioned into two new daughter cells. So let's look at each of these phases in a little bit more detail. You need to know the order of what happens, and you need to understand how it happens. So there's going to be a bit of memorizing going on here. All right, so let's start talking about what happens in mitosis. So before mitosis begins, remember we have replicated our chromosomes during the S phase. That is part of the preparation for cell division. Those chromosomes are going to get nice and condensed. Now, the reason they have to condense is that if you try to separate them when they're all jumbled up in the nucleus, things are going to get broken, they're going to get lost, they're not going to be separated equally. So part of the goal of mitosis is to be able to separate chromosomes evenly. So remember, we've replicated our chromosomes, and now one cell, or each cell, has to get one copy of every chromosome. So chromosomes condense as a way for the cell to more easily partition them. So let's talk about a new term here. We have our linear chromosome here. And that chromosome is kind of uncoiled, unwound, just available in the nucleus for gene expression. But then we're going to replicate it. Now, remember, when we replicate it, we make an identical copy. This is like a photocopy. Assuming there are no errors made, this copy should be exactly identical to the original chromosome. Now we're going to assume that it is in fact identical. There's no mutations that have occurred, and it is actually identical linear piece of DNA. When that replication happens, we produce a structure called sister chromatids. Now chromatid is a condensed chromosome. Sister chromatids are identical copies that are connected at their center. The center of a chromosome is called the centromere, and it's sort of like a waistline. It's kind of a thinner region of the chromosome, slightly more condensed. Now that centromere is going to be really important in just a minute. In preparation for mitosis, we are going to replicate our chromosomes, produce our sister chromatids, and then they are going to begin to condense into this very nice, tightly packed chromatid. Now, these sister chromatids are probably if you have ever seen a picture of a chromosome, you have probably seen the condensed sister chromatid form. And this is why we often think of our chromosomes as X-shaped, because they're held together in the center by the centromeres. This is actually two copies of a single chromosome in their condensed sister chromatid form. So keep that term sister chromatid in the back of your mind. We're going to come back to it because it's really important in just a little bit. Also in preparation for mitosis, we form something called a spindle. So a spindle is basically a little organelle in the cell that produces microtubules that are going to grow and attach to the centromeres of the sister chromatids. Now, the reason for this is that when they attach, they're going to line up the chromosomes so that they can be partitioned evenly. Spindle fibers are basically very teeny, thin structures that come from the cytoskeleton. They are similar in construction to the cytoskeleton, and I believe that I mentioned it very briefly in chapter 4, but one of the roles of the cytoskeleton is actually to produce these spindles and spindle fibers to separate DNA during DNA replication. All right, so let's get into the nitty-gritty of mitosis. There are 4 major stages in the process of mitosis. I would like you to know the names of each of these 4 phases, to include the larger phases of the cell cycle, interphase, and mitosis, G1, G2, all of those, you need to know those names. So let's talk about the characteristics of each of these stages. So in interphase, all of our DNA is sort of a jumble of spaghetti, a mess in a bowl in the center, which is the nucleus. This picture here, just to give you some reference as to what you're looking at, is actually the cytoskeleton. So we can't really see any of the other organelles. It's just the way that the cells were stained, but the nucleus is stained purple in the center. You can see some of the cytoskeleton around the nucleus, and then of course this green fibers are the cytoskeleton. Now you cannot distinguish any of the chromosomes in there because they are just loosely packed. They're not condensed at all. At this point, we've finished S phase, so we have replicated chromosomes. You can see those here with centromeres. So our sister chromatids are connected at the centromere there. So main thing in interphase is that these chromosomes have replicated. Of course, there's other things going on that's important, but as far as mitosis is concerned, replication is what's most important. These are our little spindles. So these guys are going to produce the microtubules like our cytoskeleton that are going to grow out as part of the beginning of mitosis. So our cell has now entered mitosis. A couple major things happen in the first phase, which is called prophase. In prophase, the nucleus, the nuclear envelope the nuclear membrane, that's what our book calls it. Nuclear membrane has started to dissolve. The reason is that the cell needs to access all those chromosomes. So you can see here it's not as tightly packed. We've got our spindles. They are moving toward either end of the cell. And as they do, their microtubules grow out. Eventually, those microtubules are going to attach to the centromeres of our sister chromatids. Now I want you to note here we've got blue chromosomes, we've got green chromosomes, and we've got yellow. All of these are in their sister chromatid form. Once those spindles produce their microtubules, those microtubules are going to attach to the centromere and basically tug of war until all the chromosomes are lined up like ducks in a row in the center of the cell. So now you'll notice that each spindle has a microtubule attached to one side of the sister chromatid pair. So here our blue chromosome is lined up, so one chromosome is attached there, one is attached there. Same thing with all of our green and yellow and other blue chromosome. So this phase in which all of the chromosomes are lined up in the center is called metaphase. Meta meaning middle. It is the phase where the chromosomes are lined up in the middle. If we look at our cell up here, we can see that. You'll just have to take my word for it that those are the chromosomes lined up in the middle. Here's our spindle fibers with our microtubules stretching from the spindle fiber to the chromosome. So interphase, chromosomes replicate. Prophase, chromosomes condense. The nuclear envelope breaks down, spindles form. And then in metaphase, those fibers have attached and have pulled the chromosomes to the center of the cell. The reason they were pulled to the center of the cell and lined up so nicely is so that the cell could equally partition those cells. Now, at this point, something really important happens. We have entered phase 3, which is called anaphase. And in anaphase, your sister chromatids separate. So please write that down. In anaphase of mitosis, your sister chromatids separate. They are pulled by shortening spindle fibers to either end of the cell. Ultimately, each of the new daughter cells is now going to have one copy of each of those replicated chromosomes. So because we're assuming that no mutations have occurred, these chromosomes are genetically identical to these chromosomes. In the final phase of mitosis, the phase called telophase, those spindle fibers break down, the spindles break down, the nuclear envelope begins to reform, and the chromosomes decondense. There's one little mini phase we haven't talked about yet. That is called cytokinesis. Cytokinesis is a separate phase, separate from mitosis, but it actually begins to occur at the end of mitosis. Cytokinesis means splitting of cytoplasm, and that is where the cell membrane begins to pinch in and eventually will pinch so far that it divides the cell into two. So in this image here, we actually have telophase occurring, which is the reforming of the nucleus and the decondensing of chromosomes. And we also have cytokinesis occurring, which is the splitting of the cytoplasm and all of the organelles. So the ultimate result of mitosis along with cytokinesis is the production of two genetically identical daughter cells. So for every one cell that we start with in mitosis, we get two daughter cells. Now, producing genetically identical cells is one of the key features of mitosis and the whole goal of mitosis. So now I'm going to step through this process as it looks in sequence. So we've got our parent cell, we're still in interphase there, we've got our decondensed chromosomes, our spindles, waiting to form. Interphase occurs, S phase occurs, DNA is replicated. If you look really closely, all of those chromosomes have sister chromatids. Mitosis begins, chromosomes condense, spindles form, chromosomes line up in the middle of the cell. Each chromosome is pulled apart, and then our nuclear envelope reforms and the cell completes cytokinesis. So mitosis, first step prophase, second step anaphase, second step metaphase, third step anaphase, fourth step is telophase along with the step of cytokinesis. So there was a lot going on in mitosis, so I wanted to just give you a little bit of a summary. This is important because I guarantee that I will ask you for the differences between our two types of cell division, mitosis and meiosis. So remember that mitosis occurs in our somatic cells. So this is most of the cells in our body. Mitosis produces two daughter cells for each parent cell. Those daughter cells are genetically identical to each other and to the parent cell. And then finally, something that I didn't really talk about, but is actually really, really important, and it is related to the fact the cells are genetically identical. In mitosis, cells maintain their total number of chromosomes. In humans, this is a situation called diploid. Now, diploid goes back to the idea that each parent contributes one copy of each chromosome. So di meaning two, you have two copies. Ploid means chromosome. So two copies of each chromosome, one from mom, one from dad. So remember a few slides ago when I asked you to pay attention to those blue chromosomes, the green chromosomes, and the yellow. So if you think of each of these, look at just one blue chromosome. This one maybe originally came from mom. This one originally came from dad. These are what we call homologous chromosomes. They are basically the same type of chromosome encoding all the same types of genes, but they might differ in their alleles or the versions of the genes. Now, when we replicate, we get a copy of each of mom and dad's chromosomes, so we have our sister chromatids. Then we line them up and we separate sister chromatids, and so what we end up with are cells that each now contain one copy of each of mom and dad's chromosomes. So the parent cell had two copies of each type of chromosome. We replicated all of them and then we split them in half. So we are left with each of those cells still containing two types of each chromosome. Now this is important because mitosis maintains that diploid situation. It keeps one copy each from mom and one copy each from dad. When we get into meiosis, we actually see chromosome numbers reduced and we remove that diploid situation. So when everything's working normally, mitosis occurs, cell division is completed, and your cells go on their merry way. But sometimes the mechanisms that control cell division get out of control. When that happens, we get cancer. So cancer is a situation where cells begin basically in infinite cell division. They basically become immortal. And so this can occur for a variety of different reasons. Usually, it is a mutation that occurs in an enzyme or a protein that is involved in one of those checkpoint regulations. And so basically, even though the cell has damaged DNA or isn't completely healthy, it's allowed to go past that checkpoint and continue cell division. So cancer is the second leading cause of death in the United States. It's only second to heart disease. And it is responsible for more than 20% of deaths in the United States. So cancer is a really big deal. There's tons and tons of cancer research going on. But overall, it's really hard to treat and control cancers. And the reason is that each one is different. Each cancer occurs for a different reason. And sometimes that reason is an acquired mutation. Sometimes there's a genetic predisposition to cancer. But no matter what type of cancer you're talking about, it is the result of uncontrolled cell growth. And very often, those cells also have the ability to repair their telomeres. And so whereas a normal cell would reach a point where it can't divide anymore, cancer cells can rebuild those telomeres so that they don't ever reach that odometer reading that says, hey, you have to stop. So cancer cells are genetically different. They have a mutation that causes them to lose control of the cell cycle and divide continuously. But there are a few other characteristics that make cancer cells different from normal cells. So the first is that cancer cells lose something called contact inhibition. So basically, normal cells divide until they grow and bump into other cells. It's kind of like in a city, when things get too full, people go elsewhere. Cancer cells or normal cells aren't going elsewhere, but they just stop growing. It would be as if the population in Belleville got to be too high and people just stopped having children. No more replication. I'm not saying that that is good or bad. I'm just using that as an example. However, cancer cells acquire the ability to ignore the signals of other cells around them. And so when normal cells would stop dividing because they've come into contact with each other, cancer cells don't. And they start piling up on top of each other. And this is where we get tumors. Second feature, cancer cells can divide indefinitely. So I mentioned this a few minutes ago. These cells, normally cells will reach the end of their dividing life and then the cell will die. The problem with cancer is that the cell is ignoring those checkpoints and is dividing even though it has mutations. So not only are these cells passing on the mutation that allows them to divide exponentially, but they're also allowed to divide with errors. And so not only do you start out with the original mutation that caused the cancer, but those cells begin to accumulate other errors because those checkpoints are not being regulated. They're not causing the cell to stop when they have damage. And so often in cancer cells, you have whatever mutation was the one that originally caused the cancer, but then you sort of get a buildup of other mutations that cause problems in the cells because they're not being regulated correctly at their checkpoints. Finally, cancer cells have reduced stickiness. So basically, normal cells are held together by something called adhesion. It's just the ability to stick to another surface. Cancer cells sort of lose this stickiness. And so they, even though they might form a tumor, they can also break off and move to other places because they're not sticking to like cells. So let's talk about tumors just a little bit. Sometimes cancer develops, and it develops into what is called a benign tumor. So this is a tumor that can be removed safely. It's not going to, pieces aren't going to break off, and they don't really have any lasting consequences. Sometimes tumors are removed early enough so that they don't become a problem. Other cancers just sort of form benign tumors and they can be removed safely. On the other hand, cancers can form malignant tumors. So these are cancerous tumors that have the ability to shed and spread cancer. When this happens, this is called metastasis, and you can see here that this individual has cancer in several locations. Likely that cancer occurred somewhere. Maybe this is the ovary or prostate, something like that, and it has spread to somewhere else on the individual. So what actually causes cancer to kill an organism? Well, the answer is kind of complicated, actually. It's not necessarily the tumor itself or some sort of toxic action of those cancer cells. It's kind of a lot of things that sort of come together. So as the tumor gets larger, it starts to use up nutrients and energy. And so it takes away nutrients and energy from the rest of the organism. It also takes up more space, so it presses against neighboring cells, neighboring tissues, and can kill those neighboring tissues because of the extra stress and pressure on them. And then because of that, you can have interrupted processes, so breathing, heart function, normal cell functions can be disrupted. So if the tumors are putting pressure on other tissues so that those tissues can't function, the tissue that they're growing in can't function, all of these things put together are what makes cancer so deadly. So cancer treatments are not necessarily fun. Basically, the point of cancer treatments is to kill developing cells. The problem is that treatments such as chemotherapy and radiation often kill other cells as well. So they are not only affecting the cell that they are targeted for, but they often affect other cells. Chemotherapy is generally designed to interfere with rapidly dividing cells, so it slows down tumor growth. But that means that it can slow down any other cells that are generally needed in repair and replacement. So chemotherapy often causes extreme fatigue, shortness of breath. It reduces the rate that critical cells are produced, red blood cells, things like that. And so it limits other processes within the body. Radiation is slightly different in that it is more directed at the tumor that they're trying to remove. And it is generally targeted for the tumor, but it can damage other tissue around. So wherever the tumor is, the normal tissue around it can be harmed as well. So basically, cancer comes about because of one of two different types of mutations. These are mutations that either stimulate or stop restraining cell growth. Now, these are two different things. So basically, you can think of cancer as an analogy for driving a car. So you can either, to slow down, you can put on the gas, or you can stop the gas you can put on the brakes, or you can just take your foot off the gas pedal. So there are two different ways that cancer works. One, you can think of as pushing the gas pedal harder. So these are cells that are normally dividing, but something happens to make their division go faster. So the analogy would be driving with your foot on the gas pedal. You're going, you push on that gas, driving goes faster. So the other option is basically taking your foot off the brake. So instead of, cells can also be prevented from dividing. You can think of that as the brake. But if you take your foot off the brake, your car starts to go. If the cell removes the brake, cell division happens. So either way, you can get this sort of unrestrained cell division that leads to the formation of these tumors. So there's no overall or complete cure for cancer. Many of you probably know that, particularly if you have been affected by someone in your life with cancer. But there are a lot of different types of studies going on, a lot of research on cell cycle control, on telomeres. Very often cancer treatments are designed to be effective based on the genetic makeup of the individual. And there's a lot of research going into cancer treatments and cancer cures.